Thank you, Mr. Chairman. I'd like to begin by apologizing for the juvenile diatribe from the ranking member. Science is about having an open mind. Science is about disputation, debate, evidence, and it's discussed. And the contention and the back and forth is how we determine what the truth is. But if you close your mind completely to this and are not willing to listen, I think that's how science dies. That's when we say, well, the government consensus or the scientific consensus is this. Well, that's the closing of the scientific mind, and that's a recipe for a disaster. Science is also not about ad hominem and attacks. So by attacking this panel without listening to it, leaving in a huff, and having no questions, shows a small-mindedness, a closed-mindedness, and really it should be everything that we should avoid. I'm not here to be an advocate for one treatment over the other, and I will ask some tough questions to the panel. But I'm open-minded enough to listen to their side and see and try to determine whether or not it's a good treatment or bad. But to immediately close our mind, I think is a bad way of looking at this. I had a good friend die yesterday from this, so I'm not one who's saying this isn't a deadly disease. She lived a couple doors down, and we're sad. Kelly and I are both sad about what happened. About 6 months ago, we had a friend who died in their 30s, not from the disease, but from lack of a stress EKG. She died from a terminal arrhythmia in her 30s with 2 young children, but her stress EKG was canceled because the government decided that we shouldn't do it. It was an elective procedure. It was delayed, and she died from a terminal arrhythmia. So people do also die from the decisions that people make. The most important thing is no one person is right. The panelists are not all right, and people should question them and go back and forth, and we try to figure out the truth from that. But it's a mistake to say that one person should have all the power to determine what we should do when we place too much confidence in government doctors such as Dr. Fauci. Dr. Corey mentioned that he mentioned in June about steroids. I mentioned it in March when Dr. Fauci came into my committee hearing. My first thought about the final pathway here with COVID in the end stages is it's not too dissimilar from other final pathways of adult respiratory distress syndrome. I had a friend who's a surgeon who got the strep-eating flesh from operating on somebody who had it, and a blood splatter, he got it. The chance of losing your limbs is like 50 to 70%. It's like a 50% chance of losing all your limbs. I mean, it's a devastating disease, but one of the final common pathways like COVID is your blood vessels leak fluid into your lungs. It's a sort of a toxic shock kind of syndrome. They treated him with high-dose steroids, and so I raised my hand to Dr. Fauci and said, do you think steroids might have a role in this? And he sort of dismissed me and said that the studies were showing that it probably wouldn't work. But I would think most people would argue now that one of the most significant developments in the acute care of the COVID patient has been the steroids, actually, and trying to get them the steroids before they've gotten into the full ARDS syndrome. Some are even getting it at a very early stage in the disease. But that came from people asking questions, being skeptical, and not letting one doctor, such as Dr. Fauci, say, oh, that's not right, young man, go sit in the corner. We need to have an open mind. I'm not saying I'm always right. I'm a bystander, and I'm not an expert in COVID, but I try to ask the important questions. One of the questions that I'd like to ask, and I think this is the difficulty of proving the situation on either ivermectin or hydroxychloroquine, is that because so many people get better, you know, the control group might be 99% of the people get better. Then you give hydroxychloroquine to this same group, and you might get 99.5%. It's going to be a difficult thing to actually prove in trials that you get a benefit. It sounds like in some of the sicker patients where you had a mortality, where you had a 25% mortality down to 15%, that does sound significant. Now you're getting into the sicker patients. With the outpatient studies, one of the critiques is that so many people get better naturally. How are you proving that it's statistically significant? And what comes to my mind is sort of like when we wanted to prove that taking an aspirin a day was good to prevent heart attacks. It took like 10 years and 10,000 people in the study because it was just hard to show the differences of whether an aspirin worked or not. And I think the same thing is true with either hydroxychloroquine or ivermectin in an outpatient setting, is that because so many people get better on their own. The converse is when you're really sick. because it's hard to get to treat people who are very sick. It's hard to prove that anything works when people are very sick. But I guess what I'd like to do is open it for the panel one by one to explain how we answer the critique that most of these people are getting better anyway. So how you've proven that your treatment works in a group of people who didn't go to the hospital, so that you're selecting out actually for the people who may well have been, did you prevent them from getting seriously sick, or have you selected out as your patient pool because they're outpatient, tending to be people who are going to get better? Why don't we'll start with those who are here. We'll start with Dr. Osqui, and then we'll go to Dr. Corey.
And let me quick interject, because we have such low participation. Take whatever time you have for whatever questions you have. Dr. Osqui.
Thank you very much, Senator Paul, and I appreciate your introduction. I'm not here as a politician, I'm here as a treater. I'll be very practical, and I'll defer to Dr. Corey from his experience as an intensivist. My clinical approach to patients are that they have symptoms, that I treat them early, and I typically risk stratify patients getting LDH levels, lymphocyte counts, and other blood panels whenever possible. When I've used hydroxychloroquine and oftentimes ivermectin, I've had patients go to the hospital if I didn't have an EKG or another good assessment, and I've always clinically evaluated the patients I've treated. But I risk stratify them. You may or may not know a husband and wife couple that I know Senator Johnson knows, who I treated the wife and not the husband, because I didn't deem the husband to be sick enough to warrant treatment, although perfectly safe to, but his wife was much sicker. So I think this has to be, to some degree, left in the discretion of the clinicians. In terms of the data, I provided the clerk of this committee, Ms. Kilbride, PDFs of numerous clinical trials on many of these drugs, not limited but including hydroxychloroquine, ivermectin, vitamin D, zinc, and a few other medications, and those trials are there. But the fact that we've had third world countries, second world countries that have been more innovative and have outperformed us in terms of survival, I think is something that warrants questioning. And one has to wonder if our bloated research academic bureaucracy has been more of a hindrance than a help during this healthcare crisis. But I'll turn this to whomever. I hope that's satisfactory.
Dr. Corey?
Senator Paul, I appreciate your question, and it's critical, the content of your question, which is how can we tell when something's working when many patients get better? And there's only one answer to that. And we know what that is, and it's one of the central tenets of science, which is you need a control group. You need to have a group that is comparable to those that you treat, and then you compare them to those that you don't treat. What I am trying to message today is in our manuscript, we now have 11 randomized controlled trials. Every one of those controlled trials show that in the ivermectin-treated group, lives are saved, there's less need for hospitalization, there's less transmission, less case counts. It is a fundamentally and powerfully effective therapy against COVID-19. We need the NIH to review these data. We have the data. Let me say, the amount of patients in those randomized controlled trials, in the 11 trials, total nearly 4,000, with over half treated with ivermectin. Let's remind ourselves that the treatment of COVID-19 fundamentally changed after the recovery trial was announced in June. That was a trial of 6,000 patients, 2,000 were treated with steroids, and it showed the dramatic and life-saving properties of corticosteroids. Almost overnight, the treatment of COVID-19 changed as a result of that trial. That was the recovery trial. I am presenting a paper today with more patients treated with ivermectin, with larger magnitudes of benefit than the recovery trial. I will maintain that ivermectin should be the standard of care of this disease based on these data. It's not my opinion. It's the data. We have the data. If you give anyone else a placebo based on the data that's in our manuscript, I believe that would be malpractice, and you would lead to the heightened risk of death. No more placebo is needed.
So the 11 studies are inpatient or inpatient and outpatient?
They vary. So the randomized controlled trials, I almost can't describe what this data shows. People need to read the manuscript. We have 4 randomized controlled trials in prophylaxis, each and every one highly statistically significant. patients or people, even citizens, healthy citizens on ivermectin do not get COVID.
And you're comparing it to placebo in these studies? You're considering it to this other
Yes.
standard?
So in the prophylaxis studies alone, which is 4, they took COVID-19 patients who tested positive for COVID-19. They identified their household members. They gave them ivermectin. So they had a whole group of household contacts of COVID-19 patients who took ivermectin. The other households, they didn't give ivermectin. Every single randomized controlled trial shows that in the households that were on ivermectin, drastically reduced rates of transmission. The households did not get sick. You can protect people from this disease with ivermectin. That's just the prophylaxis trials. The outpatient trials.
So the outcome tested was how many people were positive, turned up
How many
positive?
people were positive or how many people got symptoms? And they were drastically, statistically significant. These are, and from multiple centers and countries around the world. That's just the prophylaxis. We also have trials on early outpatient as well as hospital. The most dramatic is the hospital. Almost every single one, we have 4 large randomized controlled trials in the hospital, all statistically significant reductions in mortality. Dr. Reiter has a large observational control trial from Broward County, and he's here on the panel today. In his trial, he also showed the same. The patients who got ivermectin died at far less rates.
And opponents of using ivermectin, have they responded to these, critiqued these? Has there been a generalized argument that they make, or has it been silenced?
So, the opponents, again, I want to call attention to the NIH and their recommendation. I don't say that they're an opponent. They made their last recommendation on ivermectin on August 27th, which is either 10 years ago or 4 months ago. I don't know which. August 27th, this was their recommendation. We recommend against the use of ivermectin outside of clinical trials. That recommendation was based on expert opinion only. There was no data to recommend or recommend against. It was an expert opinion.
Well, I'll be happy to join with the chairman in sending a letter to try to get an evaluation. If we could go to the same question, if we've got time, do you mind? Why don't we go to the rest of the panel and ask the same question? The problem, sort of from my perspective, still being, how do you determine an outcome of something that when you do nothing, the vast majority of outpatients actually get better also?
Start with Dr. Reiter.
Thank you for allowing me to speak. So to clarify something, people talk about asymptomatic patients, but I have kind of stopped using that nomenclature. I call them pre-symptomatic patients. We do not know offhand which one of these asymptomatic patients are going to develop severe disease and become symptomatic. That is problem number 1. Problem number 2. When I put in my written testimony and also during my earlier testimony, I talk about the early intervention and treatment to decrease viral shedding and viral transmission in the home setting. And that is currently believed to be the largest numbers of people getting infected or getting infected in the home setting. So that means a 20 or 30-year-old may do fine, but now comes Christmas, there is a family meeting, and that asymptomatic individual now infects other family members, including their 70, 80, 90-year-old grandma, and now we have a real problem. What we do know about ivermectin is that it literally destroys the virus in most people within 48 hours. So hence you take somebody who is asymptomatic, but a carrier with the risk of transmission, and you essentially stop that right in their tracks. So you don't have an issue about that person now contaminating other people down the line, or you significantly decrease that risk. So that is really even a very different spin on that question that was asked. Now, to continue as far as the data, you're right, Senator, it will be difficult. My team currently has a study in place. We have the protocols written, ready to go. The only limiting factor was to get funding. I've tried everywhere. Getting the funding is extremely difficult. What we need to do is a large study, a large randomized control study to confirm the findings, to prove to the naysayers, you know what? Here is your golden standard. Now, The study that I did was a observational study, but it was propensity matched. That is the highest standard for a non-randomized controlled trial. And even there, it shows tremendous benefit. You spoke about the reduction in the overall mortality from 25 down to 15. Well, if you look at the overall mortality in people with severe lung involvement, that went from 81% down to 39%. That is even a more dramatic decrease in even these more severe individuals. It is a safe drug, a track record that is phenomenal, 3.7 billion doses administered. So I think we need to do those studies with the help of the NIH, CDC, or any one of those regulatory agencies. A study like that could be implemented within 30 days. Within another 30 days from that, we could have collected all the data. Within another 2-3 weeks from that, we should be able to have answers. If things are moved forward through the regulatory agencies, we can have our answers before this tragedy even worsens.
Dr. Balboni.
Great, thank you. And thank you, Senator Paul, for the question. I think I'll speak to the trials that we're running for favipiravir, broad spectrum antiviral targeting RNA-dependent RNA polymerase. We're running currently 2 large placebo-controlled randomized phase 3 trials in the outpatient setting, early treatment and post-exposure prophylaxis. In addition, there are 2 phase 2s, one at Stanford in the outpatient setting and in long-term care as well. There are observational studies with thousands of data points and hundreds of smaller trials having been run in many countries around the world. We need to develop hybrid trial methodologies to combine all of these traditional RCTs and observational study designs to gather real-world evidence for the regulators. If we don't pool all of these data, we're going to see COVID-19 in our rearview mirror. We're going to quickly forget about it, and all of these potential treatments are going to fall by the wayside. So I agree with you. We need to gather the data. It will be large populations, but we have to make that commitment. The only way we can make these studies work is a true public-private partnership. Single companies, as we've heard, don't have the ability or the incentive to run these trials. Favipiravir, by the way, is off patent. We're doing this at risk as a consortium. There's very little support for this kind of work. And as soon as it falls away, we've got to be beholden to our shareholders and our boards. In the meantime, as a physician, I'm trying to do the right thing for patients. We need the assistance. We need government help. This has to be a whole of government approach and a commitment to going beyond this immediate problem, which is pressing and in our faces, but to do the right thing over the long haul, to gather the data, to silence the criticisms of these kinds of studies. Multiple small studies won't work, as you correctly point out. This is what we need. Gather the data, bundle it together, and make this available for future outbreaks. And the last thing I'll say is that as part of our trials, it is true that if we were to look only at mortality data, these trials would be unacceptably large and would take too long to run. We have been working with the regulators to look at time to resolution of symptoms, similar to what was done with oseltamivir or Tamiflu. We believe that is a stepwise approach, but it's taking too long to do this. We don't have a whole of government approach to this problem. And so I ask the committee, those present and those who have decided not to ask questions, to just please internalize this message. We need a whole of government approach similar to what was done with vaccines to gather the data to do exactly what you said, which is get to these large populations to show a difference.
So next, Dr. Orion, I see Senator Hawley on the computer, so we're going to check to see whether he wants to ask questions. But Dr. Orion, you want to answer that question?
Senator Paul, that's an excellent question that certainly applies to all types of research. I would say if you have a really sick patient, in a sense you have a controlled, historical control in that patient. If the patient is going downhill and you do something and the patient gets better, then that is evidence that maybe you did something right. On the other hand, if you have a patient who's doing pretty well and you do something and the patient suddenly gets worse, then you have to be worried that you might have caused that problem. If you have a bunch of patients who are healthy and they're going to do well no matter what, then I guess you really need to have a large study, and it looks like with ivermectin they have done that. There are, of course, many confounding variables, and And then there are the ethical questions. If you have something that's going to protect your grandma, if it works and doing nothing is going to put your grandma at risk, then are you engaging in some kind of human sacrifice to use a placebo? At what point does that become the thing? Some people have even suggested that maybe the RCT is outdated and we should be looking at other statistical approaches like Bayesian analysis so that we can get good data without running into this ethical dilemma and deal with the need maybe to have combination therapies and to deal with confounding variables.
Dr. Bhattacharya.
Thank you, Senator Paul, for that question. I mean, I think the gist of your question is right. When you have an outcome that's relatively rare, what you need is very, very large samples in order to test whether and a control group to test whether the treatment actually works. You can get some indication with observational studies. There are the difficulties that the treatment is not randomly assigned. But there are statistical methods to address that. And the great advantage of observational studies is that with these statistical methods, correcting for this sort of a selection bias in the treatment, you actually can get very large samples relatively inexpensively. So there's a procedure here I think we could follow. I mean, we could use data sets like the Medicare claims data, other claims data sets, and other readily available large scale data sets to check for in sort of well-designed observational studies to see if there's some promise to the kinds of treatments that people are thinking about. And you can do this for many, many, many treatments in principle. Lots of people with different ideas can check, and then use that to inform where the large trial should be run. I mean, I think that's one procedure. There are others one potentially could follow, but the key thing I want to get across is that it is possible if you just choose to do it, and the incentives to do this have not been great, and as a result, we have much less information than we probably should have about the effectiveness of various treatments that are proposed than we should have had by this time. In part, also, I think that's been driven by this groupthink that you mentioned that has decided that there's only a small set of things that are going to work even before any of the evidence has been evaluated. I think all of that is to be regretted. I think we have to go back to the principles we know about how sound science works. We have to look at you have to do control groups, you have to do studies, but you have to have the incentives to run those studies or else they're not going to get done.